and talking about genetics and a controversial topic in social science, which is nature versus nurture. And so we're going to hear a few words of introduction. So I'm just going to introduce you to the larger Center for the Economics of Human Development that's actually sponsoring this. And here I'll just give you a few pieces of information. The co-director is here with Steve Durloff. Why don't you stand up, Steve, just for a second, or put your hand up. Okay. But the idea is really that economics, in particular social science, can really benefit by drawing on multiple channels of influence across different fields. And it is interdisciplinary, but it's interdisciplinary in a rigorous way. So the notion is not to just kind of have people meet and chat and then go back home, but actually to try to learn from each other in a constructive way. And in fact, there's specific projects on genetics underway, and we're interested in, and we're collaborating now with you, Gene, here, and with the Human Capital and Economic Opportunity. So these are some of the projects. I won't go through all this, but these are the networks that are being brought together. When I say networks, there's a larger group of people, around 450 people around the world, working on different components of this, with meetings that are often very, very technical, small meetings, and then a few general lectures like this. So here we can just talk about different partnerships that we have. There are multiple partnerships. I won't go into any great detail. But the notion is not only to develop new ideas, but also develop the next generation of scholars. So to build this process of this culture of collaboration. And so in addition to our own work, we work with the Human Capital and Economic Opportunity Group, which we're closely associated with. In fact, we direct it. So so there's no question. But this also gives us more of a global reach, but it also allows us to essentially investigate deeply. So we have a particular interest in Asia. We have the Asian Family and Transition Network, and so I won't go on too much. But we do have many different summer schools. And in these summer schools, we bring in scholars from multiple disciplines. And the idea is not only do they meet each other, but they meet economists and they meet other people in different fields, in biology, psychology, and the like. And this kind of creates what we sometimes call the Silk Road Project. Different cultures clashing, but in a constructive way. So if you want more information, you can look at this website. So we have two introductions coming. Conrad Gilliam is the Dean of the Biological Sciences Division, I guess, and a major figure in the study of human genetics. And we look forward to your introduction to Jack Gilbert, who will actually introduce Gene Robinson. So a little bit cumbersome, but here we go.
Just us between you and Gene. So I just want to comment a moment on Gene's role, as I see it, as a pioneer in building modern molecular biology and our modern biology as it interfaces with society even. So it's quite a grand vision. And this is manifested in his Institute for Genome Biology at UCIC. And here he challenges investigators to come up with a grand challenge that impacts biology and society. I mean, it's intimidating to think what he asks of these investigators, but he brings them to a center where they feel supported by infrastructure in many ways. One of the young investigators I talked to said he was challenged to put together a program project grant, had no idea how to do this, so he sat down with Gene's team and it just all fell into place. This is what we dream of doing here. It takes a lot of work, a lot of thought in this. So he's lowered the barrier for bringing interdisciplinary scientists to work together, which if you don't do that, you won't get anywhere. Everybody's very busy. But he's also done, I think, just an exemplary job of bringing, starting from honeybees, which you'll hear about today, but when you think of what he's working with today, so this is just one of the projects. I think there are 11 grand challenges, but I think this is just sort of extraordinary in its scope. So this was, at one visit, Gene announced the newest grand challenge, a $115 million grant from the Department of Energy to re-engineer the most common and fastest growing species of plant throughout the planet, and that is sorghum. The goal is to engineer them to use less water, therefore they have a smaller environmental footprint, more sustainable, that sort of thing. And they were already down to reducing at about 60% level. But also to re-engineer so these plants could, as I recall, digest cellulolignin, I believe, such that plants could, in essence, become biofactories for production of biofuels and bioproducts. Okay, so now they're bringing in agroengineering, and there were lots, we're looking throughout the planet where the temperature determinants are, where would you focus this and so forth. But then when it gets down to the molecular engineering, they'd actually put together a new startup. It was called iBioFab, and it's actually an automated means to survey, and this is probably yeast to begin with, but survey how you would, starting with biolignin or other unnatural substrates, identify the molecules that would break it down, and then using CRISPR, engineer them, get them back in, and then automate this whole thing as a company. And this is the approach to re-engineering these plants. They have fields that are under the ground where you don't see it. There are pipes. The pipes release the microbiomes are at work releasing oxygen. This is all, everything's measured in the atmosphere, everything. There are robots going down between the rows of corn. It's just amazing demonstration of team science, and that's what I thought was most remarkable. And one, you pull this together, you make it work. Investigators have several years, maybe 3 years, to come up with a major, not an R01, R01s are welcome, but major funding, and can stay as long as they need, with the understanding that they go back to their departments when it's done, okay? And it's been a major success. And there are some added attractions to this. So in addition to lowering the hurdle, Gene sort of builds a wonder of science in everything he does, and you see it in many ways. One way you saw it was they have a really nice, some of the best microscopy, high resolution microscopy you can imagine. So we were given a book, and the book was very carefully put together with beautiful pictures, but scientists took some time to explain, translate that picture into its meaning, so that it's one of the easiest books to pick up and wake yourself up in the morning when you're trying to, before you tackle something. So I get started on that, and then I'm ready to chew off something bigger. It's just amazing. Now, they didn't end there, though. They put these at the airports, they put them in the United Nations, they put them wherever people would see them. And it worked, because they also made sure that the company that made the scope, Zeiss, I believe, was featured, and so Zeiss is now supporting them. How did that happen? It was very careful planning, I suspect. So the point of all this is, for reasons that I won't go into, we're at a point in biological research where the players have changed, the approaches have changed, primarily due to whole genome DNA sequencing, internet. It's massive amounts of data. When we get a clue now, it tracks back to a single base pair, and that just changes a lot of things. It changes the scale, it changes, we need computer scientists, information scientists. With so much DNA, it's the chemists who recognize the epigenomic changes, so the chemists are now major players, both in synthetic chemists, so that we can synthesize small molecules to intervene at great precision. This now is almost a required part of biology. This puts us into medicinal chemistry. This hooks us up with the Polsky Innovation Center, because now we connect the companies. We're worried about sustaining this operation because everything seems to be conspiring against us. State won't pay their bills. I mean, the insurance rates, everything, it makes it difficult for an academic medical center, so it would be great if our science could lead out to the Polsky Center, to startup companies, and if they could be an ecosystem out there that would catch some of these startups. And Jack is throwing them out there, if they'll catch them. And then if we get feedback and just sustain this loop. And so this is what Gene is building. I sort of think he could build it better here, so we're always trying to get in here. And I think this team science or hybrid team science approach is absolutely essential to lead in the next decades. And so we can thank Gene for maybe one of 3 examples we all look to as we think of how to do this. I'll now turn it over to Jack Gilbert, who's a domain expert and can get closer to Gene. Thank you.
I don't know what I'm a domain expert in. That's an interesting term. But it's my great pleasure. I first met Gene when I was trying to build the Microbiome Center at University of Chicago, and I went down there to actually see how you should run an institute. And he's done an incredible job at building that. So I will mirror everything that you've said, and I think that's absolutely brilliant. One thing, so this Carl Woese Institute for Genomic Biology is a phenomenally functional program, a phenomenally functional thing that works and integrates into a university environment which is supportive of it, and I think that's wonderful. However, bear in mind, this guy's a bee guy, right? Apis mellifera being your organism of choice, he's obsessed with behavior, obsessed with manipulating the characteristics of how the system should run. And I've got colleagues of mine that have worked with him for years that say, you know, I just feel like I'm just part of a big experiment. Gene is just manipulating the sociology of this environment to see how to make us more productive. But it is true. If you look at, I got this quote here that, you know, your research focuses on the division of labor, aggression, and the system of symbolic communication. Which is perfect faculty. You know, essentially, yeah, you can figure out that, you could solve all the problems that we have. It would be absolutely amazing. Gene's kick ass, he does great work, blah, 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 blah. He just won the 2018 Wolf Prize, which I think is great for agriculture, for his work in the genomic revolution of organism and population biology of the honeybee. I love the honeybees. And they're dying at the moment. We need more honeybees. And it's because of microbes. So the microbiome of the honeybee is the most important thing. Screw the genetics, sorry. The best quote, though, that I come up with for you, you had a science paper, just a perspective on the principles of what you're going to talk about today. And the opening quote was a Spanish proverb about the horns of the dilemma are always on the same bull. And I think, you know, again, you can learn a hell of a lot from that if you can figure out why you have a dichotomy, why you have a problem and a solution. Figure out what the root of that system is, and you can change the world. Without much ado, Gene.
Well, thank you very much for all of these introductions. I'm just really,
We've got 5 more.
I'm just stunned. I really appreciate it. At the Institute, I don't need to say this, but I will say this. We all stand on the shoulders of others. There were many people that have worked to create the Institute of Genomic Biology. I've been really privileged to lead it for the last 7 years, and it's an extraordinary set of people, faculty, students, and postdocs who are interested in this extreme team science approach, and we are able to have a really extraordinary group of staff who can support this at a very high level. As Conrad said, everyone leads a double life. They're expected to continue working in their home departments, and so to make that possible, we have to lower the activation barriers to be able to do that. And that allows us to be very, to use Jack's word, aggressive in our evaluations, so that we really stay at the cutting edge in the Institute and keep the teams functioning. The idea very much, I didn't know about the manipulative part, but the idea very much is a beehive, which is that the whole is greater than the sum of the parts, and that's what we look for. So I'm really honored to be here. I've had several wonderful conversations with Jim that have led to this invitation, and we are very interested in joining with some of the amazing work that's going on at the center here and trying to put in additional biological perspectives. It's also great to see so many people that I know, and speaking of the IGB, in a past connection, an IGB fellow started her career as an IGB fellow. Professor Sarah London is here on your faculty, and I'm delighted to give her a shout out. And also to kind of put in context, a lot of the biological work that I'll be talking about does draw upon my beloved species of choice, the honeybee, but there are other species that are really contributing to this exciting moment that we're in right now, which is to realize the impact of discoveries that have been made over the past 15 years on the dynamic genome, as I'll call it, the idea that the environment affects genome activity. And we need to now, I think we're at the stage now, especially with human biology, to be able to address the questions of what are the implications of that for understanding human behavior. So Sarah works on the songbird, and that's a prominent species in Socio Genomics. And I won't be talking about any songbird work in particular, but just keep in mind there are a number of species that have been used to build the synthesis that I'll be talking about. So I want to talk about Socio Genomics and the dynamic genome, and give some thoughts on perspectives on how this relates to nature-nurture. In my first 2 slides, I will telegraph the punchline of the seminar and what the really important points are, and then back up and talk about some of the science that leads to that. But pay attention to the first couple of slides. That's really the main messages. So Socio Genomics is a term that I coined about 15 years ago, and the two foundational pieces can be seen here. First of all, social behavior is influenced by genes and shaped by evolution. In other words, E.O. Wilson in 1971, that's a mistake, 75, had the book Sociobiology, which basically made the argument that social behavior has a biology. If something has a biology, that means two things. It has a mechanistic basis, a set of biological mechanisms that regulate it and give rise to it, and it has an evolutionary history. So that's one foundational piece, especially the evolution piece, because the second foundational piece, which came out of the field of Evo Devo, and Sean Carroll as a principal investigator and explainer in that area does a beautiful job of talking about gene function being conserved between vertebrates and invertebrates. So those are two foundational pieces. Genomics as a young science is what has made this all possible. Genomics is about 40 years old, and a contribution of genomics to this foundation that perhaps could be on this slide, but is really more generic, and that is that genomics has taught us that we all play with the same deck of cards. That is, all organisms share an unbelievable amount of genetic material and are playing with the same genes. Not all, there are species-specific genes in all lineages that have been studied to date, but there's a surprisingly large foundation of common material. That wasn't known before genomics started. So those are the foundational pieces of sociogenomics, and I want to now talk about three principles of sociogenomics that come out of the animal studies. I mentioned that this is an exciting moment, and by that I mean that some of this work has been going on for about 15 years, and the body of work is getting stronger and stronger in the animal world. And now, as we get to the point where we're really thinking about large-scale human genome sequencing projects, that is, countries are already, if they haven't already started, they're thinking about sequencing the genomes of all individuals in their countries. Medical systems are doing that. So we're at a moment in the study of human behavior where the roles of genes and the environment are going to be playing new roles, have new implications, and we now have an obligation to look at this body of work that's coming out of animal models and what can we learn from it? How do we frame the study of human behavior? I teach genes and behavior, not too often nowadays as I run an institute, but I usually, to get the students' attention and to make an honest point, begin the course by saying that behavioral genetics is one of only a couple of fields of science that has done incredible harm to people. That's a legacy of shame that we have to live down. We're now at a moment with genomics that we can get it right with respect to understanding human nature. And the body of work has matured. The interest in bringing genomics into humans has matured, has deepened. So we're at a really interesting convergent point that gives, I think, new impetus to trying to bridge these gaps that we have tried to bridge for many years, but which genomics, and I will assert, with the sociogenomics paradigm, gives us an opportunity to do so. So I'm going to talk about work that supports these three principles. Number one, brain gene activity is closely linked with behavior across timescales from physiological to evolutionary timescales. Those of you who are biologists in the audience know that we usually talk about brain gene expression rather than brain gene activity, but I try to pitch this at a broad scale. Secondly, environmentally induced changes in gene activity cause changes in behavior, so there is a causal link from that direction. And then thirdly, something that echoes what you just saw, the relationship between genes and behavior is highly conserved from animals to humans. And so these three principles then give rise to a simple statement that because of the dynamic genome, because of this environmental regulation and input, DNA is not destiny. And this then has a number of implications for understanding human behavior, including the notion that if there's a malleability of human traits, what are the policy implications for that? These are the kinds of discussions that Professor Heckman and I have started having, and which we're both so excited about and love to get your feedback on today. So just to project that a bit more before I go into the scientific background, I said this in a different way, and now I'm saying it here. We're really poised for a new understanding of human nature. So there are studies coming out in the human realm that are already starting to build on this dynamic genome possibility. Brent Roberts, a psychologist who has already been collaborating with Jim, has taken the Socio Genomics model to develop a new model for personality traits, which deals with the malleability of personality, despite the fact that it has high heritability. So that's one strand of thought, of study. Another one pioneered by your colleague who sadly just died a few months ago, who's a wonderful colleague to me and a towering member of his profession, John Cacioppo, studying social regulation of gene expression in human leukocytes, and the idea being here that the loneliness feature that he studied so much has a genomic imprint on it, and I'll come back to that towards the end of the talk. Behavioral genetics is starting to reach into the court, and there are several cases that have admitted evidence related to individual genotypes with respect to a couple of genes, but in particular a gene that has a known genotype by environment interaction. That's the only one that's gotten real traction in the courts. Some of you know this, of course, as the MAOA gene, which has an environmental component to its behavioral manifestations. And then lastly, a paper that just came out, paper by a social scientist, Behavioral Epigenetics, the last nail in the coffin of genetic determinism. So if you just remember this, that's kind of what I'm trying to talk about. And so some models, I led an effort in the National Academies to have a study section on, you can't quite read it, it's called Biological Embedding of Early Social Adversity from Fruit Flies to Kindergartners. Genomics allows us to say that now with robust biological information. It's not a reach any longer. It still is a reach, but it's starting to be, we can substantiate that kind of connection. And this is from some of Brent Roberts' work, a new model for personality that brings environment and DNA epigenetics into register with some traditional personality development concepts. Okay, so where are we right now? Well, I think we're at this interesting moment with respect to the public's perception of genes, and this relates, of course, to where we're all going to be going with human genetics. We have no problem thinking about heredity or thinking about the role of genes when we think about sports. These are the Manning brothers and their dad. We see that, we kind of nod our head, and it just sort of feels right, and we move on. But when we get into traits related to educational attainment, intelligence, personality, social behavior, there's a lot of discomfort that people have. And this really stems from the idea that there still is a notion out there that genes in the environment are separate. It's like apples and oranges, and we think about them separately. We in biology are enlightened because we know it's not nature versus nurture, it's nature and nurture, but we still use this construct of genes and environment to suggest that they're really something different. And that, of course, genomics has taught us, and applying genomics and sociogenomics has taught us that that's not the case. So here's the model that I think many of us are working with, and I want to show you some of the data that support this. So here we have the traditional Holy Trinity, appropriate for a room that looks like this, in molecular biology, DNA to RNA to protein. The biologists in the audience, of course, recognize this. Some other ways of looking at this are that the genome is composed of DNA, and then when the genes are turned on or turned off, different places, that's how you go from DNA to RNA, and all of that collectively is called the transcriptome, ome being a word that we just add for all, everything, and so all of the genes that are transcribed form the transcriptome, and then protein is the work part of the whole thing. So again, something that I explained in my class is that genes are boring when you understand what they do. They just encode proteins. That's what they do. There are no mean genes, no smart genes, no dumb genes, no aggression genes. Genes just encode proteins. Proteins work together in incredibly interesting ways that we're only beginning to fathom to create networks that then build the body, build the brain, repair the brain, allow it to learn, and so on and so forth. And so proteins then are the basis for the higher level traits, or in the jargon we call them phenotypes. So these are the constructs, and the big point of sociogenomics is to recognize that the environment plays a big role and affects the genes when they're turned on, where they're turned on. So this is the biological embedding piece. We don't understand it. We're just beginning to understand how the environment gets under the skin, is sensed by neurons, travels through a sensory system, is processed by the brain, gets inside the nucleus, inside the cells in the brain to orchestrate changes in gene expression that then allow for changes in behavior. So that's really where the action is. But this is the new piece, and if you recognize this, then you see that genes in the environment, yeah, but it's all about, as Jack said, that quote, the horns of the dilemma are on the same bull, the bull in this case being genomes, hopefully not the talk that I'm giving right now, and the idea being that you have hereditary and environmental influences on the genome. Hereditary influences are long-term, from generation to generation. That's what we call nature. And then environmental influences are more short-term, minutes to hours to days to months to even years. And then even some now increasing evidence of from generation to generation coming from the environment. And so we have two distinct influences on the genome, but where the action is and where it's integrated is at the genome level. So how did I come to all this? I fell in love with honeybees 40 years ago, and most biologists fall in love with a question and then find the right organism or system to study that. A small minority fall in love with an organism and then try to figure out how to make a living at it. And so I'm in that small category. Fell in love with honeybees 40 years ago, quickly realized that the way to make a living with them is to appreciate and understand their social organization. So they live in colonies of about 50,000 individuals. Their brain is the size of a grass seed, but collectively the whole is greater than the sum of the parts, and they're able to change behavior, reallocate labor, communicate symbolically. They are one of the most important organisms on the planet because of their pollinating activity. They amount to some $15 billion a year in pollination activity in the US alone. Many crops would not be available without their pollinating activity. They do things on a grand scale. They're overachievers. And the reason they're overachievers is because of their bonds, their social bonds, and their coordination of activity. So for today's purpose of today's talk, the key feature is that because of this division of labor, there's specialization like we have in our society. So there are individuals that have different roles for extended periods of time. Most animals do lots of different things, but they pass through those states very quickly. And so there are genomic components to those states, but in some cases they're harder to find. This is a classic model system. We have exaggerated behavioral states. Social evolution has given rise to these distinct behavioral states that go on for days, even weeks, and in a lifespan that's just 6 to 7 weeks. So they're exaggerated states that provide easier material to be able to study at the genomic level. So these are some of the categories of individuals. Many of these relate to a behavioral maturation process, which I'll mention again, but the idea is that bees start by working in the hive, and here's a cartoon. An adult worker honeybee lives 4 to 7 weeks. She spends the first 2.5 to 3 weeks working in the hive, then she graduates to working outside. And so some of the jobs that go on inside are taking care of the baby bees, the nurses, building the combs, so the builders, and then a variety of activities that go on outside, foraging, scouting for new food sources, that'll be important later. Guards and soldiers are important, and, I got scouts twice, only one group of scouts, sorry. And then corpse removers, there's a group of individuals that specialize on removing corpses, and even more. The key features experimentally of the system that commend it are that, they're insects, they're small, but they are big enough to study on an individual basis. And nowadays we're using barcodes, so we can barcode every single bee in the hive. We do long-term monitoring. We can get all of the individuals sampled all the time to know what they're doing. We're not completely at that stage yet because you have to build the behavioral identification modules one behavior at a time. So kind of a secret that I just found out that I'm not too pleased about is that machine learning is not as advanced as I thought it was anyways, because we can train an undergraduate to identify all 25 or so behaviors in 1 week, and it takes the machine learning algorithm about a month to learn one behavior. So maybe there's some expert in here that says we're doing it wrong, and I'd love to hear from you. But we're working on the system. We have several behaviors that can be monitored. So the individual level behavior is easily observed, and yes, it is manipulable. And we can do that because the social environment can be precisely controlled. So each of those stacks that you see here right there is a beehive. 50 to 60,000 individuals. As you can see, we can keep a lot of them there. So we can manipulate things at the social level and then study it at different levels of biological organization. So we're interested in how the environment affects the society as a whole. We're interested in how the behavior of individuals give rise to a smoothly functioning individual. Then we can get inside the individual, study its brain, and understand how neurons and genes contribute to behavioral plasticity, social roles, and study that in that way. And so we can go from society to genes, from genes to society. These different levels, they are accessible to biological analysis. Some model organisms are way more accessible at certain levels, like fruit flies, for example, way more powerful at the genetic level, but there is no social level. There's no society that you can study. And so the honeybee gives you a level of accessibility that is nice because of its breadth all the way from society to genes. One example, which I'll talk about, is we can do cross-fostering without filling out IRB forms, I might add. So we can take individuals from one society, put them into another society, characterize those two societies, and then do those kinds of experiments, classic common garden experiments, and so forth, in situ, in a social environment. Okay, so that's the general model system. For today's talk, I'll be talking about data results that come from two behavioral systems within the honeybee colony. One is Hive Defense, so I want to motivate that, and then motivate the second one, then we'll move on. So bees defend their colony. They defend their colony because an unintended consequence of their success is that they are vulnerable to predation. So bees are consummate vegetarians. They live exclusively on flowers. They collect nectar and pollen. That's what they eat. That's all they eat. 20,000 species of bees on the planet. They're all vegetarian except for one that was discovered to feed on carrion. So it's an extreme form of specialization. Nectar is a form of food that is highly dilute, so it is sparse in time, sparse in space, and it's hard to make a living. The way the bees have done this is they use social foraging. They focus their activities, they talk to each other, they communicate, they work really hard, they collect large numbers of samples of nectar, and then they bring it back to the hive, evaporate the moisture, and turn it into honey. Nectar is 45% sugar, honey is 85% sugar. I ask you, if you're a predator, or someone looking for a meal, like Winnie the Pooh, and you're contemplating what to eat, would you like to go from flower to flower and get little microliter samples of 45% sugar, or would you like to knock over a beehive and get 100 pounds at 85% sugar? So their success at turning a poor quality food source into a high quality food source makes them vulnerable, and so they have evolved protective measures. These protective measures involve hereditary influences, so places where there are more predators, there are higher levels of aggression in defense observed. Social influences, there are groups of bees that specialize in coordinating. Bees at the entrance act as guards. You can see their behavioral postures. Their front legs are off the ground, their antennae are facing forward, and they act as the bouncers in the hive. They can recognize who lives there, who belongs there, who doesn't, by means of olfaction. And so if they detect that someone's trying to get into the hive that doesn't belong, they release an alarm chemical, alarm pheromone, that alerts others. The group of bees most alerted are the group of soldiers, who are the first to fly out and attempt to sting Winnie the Pooh and others and try to deter the whole process. So you have social influences that are tuned by environment to create hereditary differences. We exploit this with a strain of bee that will be called AHB, that's more aggressive relative to a strain known as EHB. So you'll see that. So that's model system number one. Model system number two, and then we're finished with the preliminaries here, and we'll get to it, are the scouts and recruits. So I mentioned that they're dealing with a low-quality food source, and one of the ways that they handle this is they group together. And so there's a division of labor even among the foragers. So there are two groups, scouts and recruits. Scout bees go out, seek food independently, and then they communicate their findings to the other bees. Most bees in the hive wait, and they wait to be told what to do. They wait to be told where to go by the scouts. So the scouts seek food independently. If any of you study animal behavior and you work with animals that are more sort of solitary in their life, at this point you'll say, that's no big deal. My animal goes and scouts, finds food independently. True, but then look at the second one. Scouts abandon profitable food sources. They're the young and the restless. They're always looking for the next food source. This one should grab your attention because this violates a basic precept of behavioral ecology, which is win, stay, lose, go. Most animals continue to do something until it's no longer profitable to do that, and then they do something else. Scout bees find a good food source. They communicate by means of the dance language, which is symbolic movements and sounds. And then once there's a group of bees that are recruited to that and collecting food from that, they go on and look for a new food source. They show novelty-seeking behavior, and just to give you a spoiler on that, those are the kinds of genes that we find that distinguish scouts from recruits, genes that have been previously identified in studies of vertebrates, including humans, to be associated with novelty-seeking. Scouting, like aggression, also has heritable and environmental influences. So some bees are more inclined to be scouts, but depending on the environmental conditions, that can be tuned. So if there are many resources available, the number of bees that are scouting goes down. And if it's very hard to find food, the number of scouts goes up. So there's some extreme scouts that will always scout no matter what, and that seems to be hereditary. And then there are others that are more situational. They're more on the border, and they can kind of flip either way. Okay, so brain gene activity is closely linked with behavior and across time scales. So I'll show you data from our work on aggression to support that statement. So what you see here is just a way of clustering the activity and the patterns of different genes. So I'll be mostly relying on, so I want to take a moment, I'll be mostly relying on studies that look at large-scale measurements of gene expression. Transcriptomics, as it's known in a jargon way. And there's a variety of platforms that have been developed. Right now, the current one is you actually sequence. Remember, those of you that just learned the DNA, RNA thing for the first time, remember RNA transcribed, DNA transcribed to RNA. That's what the genes gene expression, gene activity is all about. So you can measure that gene activity. Right now, the method of choice is to actually sequence the RNA and see how many copies of each gene are there, and then develop a profile of gene expression profile. Then you use bioinformatics to try to derive meaning from that. One simple thing you can do is cluster genes by virtue of their expression pattern. And so you get these genes are showing similar patterns of expression, and then when you compare across behavioral states, you see very different patterns, the false color giving you measures of upregulation or downregulation. The statistical measure is differential gene expression. You get a list of genes, differentially expressed genes, DEGs. You'll see the term DEGs. Here and there. So here's one such experiment. We're looking at comparing the aggressive strain of bees from a less aggressive strain of bee, and we're looking at 3 different behavioral groups: guards, soldiers, and foragers. There's a great deal of difference between the strains of bees in their defense, very little difference in their foraging, and this largely tracks what you see in terms of individual genotype contributions. You see that larger numbers of differentially expressed genes in the brains of guards and soldiers relative to foragers. That fits. We did then also an experiment where we exposed bees to alarm pheromone. We see a large number of genes showing a response to this. And these are some of the foundational studies. I'm going to make some points in a second, but one thing you can do, you can see such big differences in state that you can then use supervised, unsupervised methods. This was a supervised method. Leave one out for those of you that do this sorts of thing, to make predictions and ask how many genes does it take to be able to predict behavioral states. So you can turn this into a predictive sort of thing when you have differences that are as extreme as the kinds of things that you see here. So behavioral states have an associated brain gene expression profile, and you can get these, and you can compare these states in a number of ways. And I'll show you some of the conclusions that you can get. First, one conclusion is the picture you should have is that these genes are kind of turning on and turning off constantly. So DNA might be a script, but there really is an improv going on. The environment is affecting the expression of the genes, and so this is meant to simulate or to evoke that for you. So alarm pheromone causes hundreds of differences in brain gene activity. It was in the 430 range, was one conclusion from that slide. Another one is that I didn't touch on that slide, but I just want to make the point here. We did a cross-fostering experiment, as I alluded to, put AHBBs in EHB colonies and vice versa, allowed us to do a very nice, simple two-way ANOVA on the influence of the individual genotype and its colony environment on that. And when you do that, 25% of the influence or on the variance of gene expression is due to the colony type. So strong effect of individual, but 25%. So this is nurture right here, 25% of the variance affecting this. Then the other point that I mentioned is that you see these changes in at this point we have to say, surprisingly or perplexingly, intriguingly, coherent way across timescales. So when we look at the DEGs, differentially expressed genes, so we have a list that distinguishes the aggressive strain from the less aggressive strain. We have a list of genes that are responsive to alarm pheromone. When we compare those two lists, there's a highly significant overlap. So genes that are responding in real time to an alarm pheromone have been tuned over evolutionary time to the different ecologies that these two different strains evolved in. This was work that was, almost about 10 years old. I said this whole area is about 15 years old. Here's some recent stuff, more recent stuff, that puts another layer on this that is really what's helping us understand the environmental influences, and that is epigenetics. Here's one particular kind of epigenetic marking, and that is the addition of methyl groups to DNA, methylation. This is just really technical stuff on what particular combination of nucleotides in the DNA is methylated. It's not important for today. What is important is big differences between the two strains of bees, AHB and EHB. So if you look at the brain, you see that there's a lot more methylation of genes in EHB than AHB. The function of methylation is still very much in flux. It has a number of functions. We don't know in particular which one is driving it. One function is a dampening of gene expression. That's primarily seen in vertebrates, where the methylation occurs in the part of the gene that regulates when the gene is going to be turned on, the so-called promoter region. Other places of the gene that's methylated have to do with what version of the gene is going to be expressed. Some genes come in different flavors, and they can be alternatively spliced, put together in different ways to create different forms of a protein ultimately. So that can be an effect here. But I want to make a more simple point for today, and that is big differences between AHB and EHB. And then again, another overlap story. If you compare the genes that are differentially expressed, this is all in the brain, by the way, between EHB and AHB, and the differentially methylated regions, you see a significant overlap. So epigenetics, in other words, figuring into this environment affecting the transcriptome in this particular way. That methylation is involved. Okay, point number 2, environmentally induced changes in gene activity cause changes in behavior. So we've established a connection. What's the functional significance of this connection? So I'll continue with aggression on this to make the point that these changes in gene expression actually provoke changes in future behavior. So let's step back for a moment. So these experiments can be done, this one makes a really good point right here. 30-minute intervals, this is behavior, but it reminds me to tell you this. We do these experiments, we expose an animal, set of bees, to a particular stimulus. We measure the changes in brain gene expression sometime after that. 30 minutes, 60 minutes, 120 minutes, we see these robust changes. The animal responded immediately to the challenge. Animals respond to survival-based behavior faster than changes in gene expression can occur. So what are all these changes for? I wrote a short paper that basically argued neurons are for today, genes are for tomorrow. The idea is that there are two control systems in the brain, and neurons are able, they're tuned, they're ready to go, neural systems are ready to go, they respond to stimuli and deal with it. But in many cases, the exposure to one kind of stimulus, ecology of that animal will predict that there's going to be other kinds of exposures. So for example, threatening behavior, for most species, a threat, a particular challenge, usually means there's going to be another challenge. So the neurons respond immediately, but then through ways of biological embedding that we don't understand yet, the message gets through to the nucleus in the neurons that, oh, you know, there was one threat today, there's probably going to be a threat tomorrow, and it tunes the system so that they become more vigilant. So here you see behavioral data that make this point. If you expose honeybees to alarm pheromone, you will see an increase in colony responsiveness measured by the number of bees flying out and trying to sting us. And yes, we get stung often. And you can see that this kind of levels off after 90 minutes of response. Here is the same thing in a laboratory behavioral assay, different number of colonies. And so here we have a bifurcation where we have the exposure to an intruder in a laboratory assay. Some bees respond aggressively. There's some that don't respond as aggressively. We can note that. And then we look at their response to a second intruder. Those that responded aggressively the first time are much more likely to respond aggressively the second time. Some of you that understand this kind of work will realize that we're confounding hereditary and environmental influences, and in fact there are both. But when you look at these two together, you can see that there is an influence of your past performance on future behavior. Now, for those of you who are hobby beekeepers, you know all the stuff that I just said, which is if you open up a beehive, leave it alone for a couple of hours afterwards. Don't go back to it. But we proved this scientifically. So one of the changes that we saw in brain gene expression pointed to a profound change in brain metabolism. Won't go into all the details, but many genes have known functions, and that allows us to tell not just if the gene is increasing activity or not, but what it represents in terms of the biology. And in the case of exposure to social challenge, what we see are that changes in gene expression to cause a big change in brain metabolism, how they're turning fuel into substrate, basically, how they're burning sugar, how they're burning their energy. And so the nice thing about finding this is that there's a lot of understanding of metabolism. It's one of the most well-developed areas in biochemistry. Reactions are well known. We have ways of manipulating metabolism to be able to test for a functional connection. So I'll show you data from two species, honeybees and fruit flies, to make the point that if we change the brain metabolism in the way that if we experimentally change it in the way that it occurs naturally when bees are exposed to an intruder, we can increase aggression. So with bees, we used pharmacology, and with Drosophila, we used more state-of-the-art genetics. So bees that have an injection, this is the work of two former postdocs who are now independent faculty members, Hongmei Barilai and Claire Richoff. If we inject pharmacological agents into the brain to mimic that change in brain metabolism, for those of you interested in metabolism, an increase in aerobic glycolysis, a decrease in oxidative phosphorylation, we can increase aggression in a dose-dependent way. This is the behavioral assay, 10 bees in a dish. We put an unrelated bee in. As I said, they can tell an intruder, and then a large number of them will react aggressively. And when we do this with bees that have been treated, we get a level of aggression that's above the baseline, which is the control here. For Drosophila, we use a genetic approach. And we engineer flies that have a change in brain metabolism by manipulating this gene, the ND20 gene, which is involved in oxidative phosphorylation. These are the two parental lines that are used to create this genetically engineered fly. The parental line, this is where the gene comes from. This particular line allows the gene to be expressed only in the brain to get more specificity. This is a much more refined approach than just injecting into the brain pharmacologically. biologically. This, in fact, we were able to do this expressing this particular construct just in neurons or just in supportive cells in the brain, the so-called glial cells, to be able to get more specificity. Regardless, either way, you see an increase in aggression. This was pleasing too because honeybee defense is in a social context, defending their hive as represented by however they're transposing that in the little plastic dish. In flies, it's very different. They're competing as individuals for resources related to mating. So these are male-male interactions, and we're counting the lunges. So aggression in a very different context, but the brain system is engaged. Similarly, we've seen similar things in stickleback fish and mice. Changes in brain metabolism associated with an exposure to a challenger. Again, one of these common themes that I mentioned. Okay, and then finally, the relationship between genes and behavior is highly conserved from animals to humans. So back to the Institute. This is one of the multidisciplinary teams that Conrad spoke about, and this team is the team that my lab is privileged to work in, gene networks in neural and developmental plasticity. We were able to develop a large-scale project, get funding from the Simons Foundation to be able to ask this question in a very robust way with 3 different experimental models. Lisa Stubbs is a mouse geneticist, Allison Bell studies stickleback fish, neurogenomics, and then my lab in honeybees, and then Dave Zai and Saurabh Sinha are computational genomicists to help us derive new meaning from these data. So here's the question as it's posed, and it should be a familiar one by now in this talk. Social information processed by the brain involved in some way in a behavioral change. Are those molecular mechanisms, molecular pathways, are there conservation there in some of the pathways that allow animals to do this kind of thing? And the answer is yes. So I'll just give you some highly processed data, just one slide. So we do the experiment, I'll back up, we do the experiments, we find the differentially expressed genes, the DEGs, and then we can compare them, and yes, there's a highly significant overlap across the 3 species, and we've done a variety of things with that. One of them is to look for a particular set of genes that are very important in orchestrating changes in gene expression, transcription factors. So I mentioned epigenetics as being important, but there are a set of genes that are important in orchestrating gene expression changes. These are the transcription factors. Their job is to turn other genes on and off. They work in combinatorial way, but fundamentally you can establish relationships between transcription factors and their target genes. So when we do these overlaps of genes differentially expressed, focusing on transcription factors, we find a set of genes, set of transcription factors. We're using the mouse gene names as the more well-known names, but we find very robust orthologs, which is a technical term for genes that are so similar in sequence, despite their being in different species, that you can, using statistics, label them as orthologs, cousins, if you will. When we do that, we see that there are some transcription factors that are involved in orchestrating changes in gene expression in response to a social challenge across same transcription factors across very distinctly related individuals. So that's the case for animals. Can we go further? That's why we came here today, right? Well, to start this off, here's a quote from sociobiology. This time I got the year right. It is the task of comparative sociobiology to trace these and other human qualities as closely as possible back through time. So E.O. Wilson had that vision, but this was beyond science's ability in 1975, and as many of you know, that caused a lot of problems, that assertion, with the inability to actually prove that. But we had a serendipitous situation with our honeybee studies that led us to address this question. This is now the work of Hagai Spiegler, a postdoc in the laboratory. So Hagai did all of the work on social challenge, exposing the bees to an unrelated individual in the laboratory, the stuff that I just talked about, the bee, mouse, fish stuff. I didn't tell you that we did 2 sets of experiments, one on social challenge, the other on social opportunity. So a negative valence stimulus and a positive valence stimulus. For mouse and fish, the positive valence, the opportunity, was a mating opportunity. For honeybees, it was a rearing opportunity because worker honeybees don't mate. They don't generally lay eggs in the hive. That's the queen's job. The worker's job is to do all of the supporting activities, and their reproduction occurs through the closely related queen. So Haggai conducted these experiments. The challenge with the intruder, the opportunity was to give, again, a dish of bees a queen larva, because it doesn't get any better than that if you're a bee to raise a sister to become a queen. So that was the social opportunity. So when Haggai did the challenge assay, he noted some bees responded strongly and some bees didn't respond. And we said, yeah, that's behavior. You see that variation. And we proceeded to study those that did respond. Then 1 year later, he did the social opportunity assay, and he found some bees responded and were avidly nursing their sister and trying to get it to grow up to become a queen, and others were just hanging back and not responding. The two things were done 2 years apart, but it led us to ask, well, might there be some unresponsive bees, bees that are just not responding to stimuli? Because that would be an interesting observation. So he redid it using this combined assay now, presenting both stimuli and then asking the question, are there bees that don't respond? And the answer is yes. Here's the ethogram. So these are bees that are responding in different ways. So the color coding is the guard bees are red. They are responding strongly to the challenge, but not to the opportunity. The nurse bees are blue, responding strongly to the opportunity, not the challenge. We had a couple of hyperachievers that were responding to both. That's a kind of an aberrant situation, actually, because of the strong division of labor. What we did see that, but we also saw about 15% of the bees on average across, these are different colonies, different genetic stocks, unresponsive to either of those. So is this a state? Well, we used transcriptomics to ask that question. So we compared brain gene expression. Now we're working in a particular region of the brain that we implicated in the previous studies, which I didn't mention, the mushroom bodies. For those of you studying insect brains, I know there's some great Drosophila folks here today. We used the mushroom bodies and measured brain gene expression in the mushroom bodies and found big differences, 1,000 genes differentially expressed between the unresponsive bees and either the guards or the nurses. This DEG list allows us to do, this is a higher treatment of the data, this is a principal component analysis, just to make the point that the individuals more or less cluster separately. So different colors are two different genetic sources, or colonies, and the blue bees, the nurses, are mostly together, the red bees are mostly together, and the green bees, the unresponsive bees, are mostly together. So this looks like a state. Does it make sense at all? Well, it appears to make sense from the perspective of behavior in the hive, because in another line of study in my lab using the bar-coded bees, we have been creating basically a Facebook for bees, and that was a much more popular thing to say a couple weeks ago than now, but be that as it may. So we have a camera here. The lighting, you can't quite see everything. Everything's done under red light. Bees can't see red light. There's a camera here that's aimed at the observation hive, so that we get an image every second. Here's the observation hive. These are the bees that are bar-coded, and you saw the inset right here. So we get an image of every bee, and then in this particular case, what we're looking at is how connected they are to each other. There's a particular behavior known as social feeding, where they're touching each other's antennae and they're swapping materials. It's not strictly nutritive because it's actually going both ways. Those of you that know primate grooming, it's sort of like that. It has a lot of social meaning. It's freighted with social innuendo and so on and so forth. And so we use that as an indicator and built this social network. And when we do that, so here's the queen. After 1 week, the queen had 745 interaction partners, friends, and then 4,368 interactions. So that's just to give the ground truth. She had the most, so the system's working. We just published on it, validated it, and so forth. Be happy to talk with you about this. But my point here is that there's a great deal of variation between individuals. Some bees have almost as much social interaction as the queen, but here is the point that I'm trying to make with respect to what we're talking about. There are some bees that have hardly any interactions. 1 week, 24/7 continuous monitoring. Some bees have 500, almost 1,000 interactions, and here's like 24 interactions. So the idea of seeing socially unresponsive bees in the laboratory doesn't seem like an artifact. They could be fitting in with this profile here. Did you have a question?
What are the bees gathering for doing this
Yes, so these are hives that the bees can go out freely, go through a hole in the wall. Yeah, thank you. So we took the DEG list then and compared it to genes that are implicated in autism. Now, I want to emphasize very, very strongly that autism is a very complex human syndrome with many different components to it. And I in no way am trying to simplify this or speak in a very facile way. Bees are not humans. But one component of autism is social unresponsiveness, and we have a manifestation of that in the bee, and so we explored it on that basis. So let's be clear about that. There are two sources of information on the genetics or genes involved in autism. One comes from DNA-based studies looking at variants, and this is funded by the Simons Foundation. It's the Safari database. And then the other comes from gene expression studies of postmortem brains. We compared our DEG list, our list of about 1,000 genes that distinguished the unresponsive bees from the guards and nurses, to the gene lists available through these studies. And what we found that for the higher confidence genes in the Safari database, there was a significant overlap of the genes on our list and the genes on their list. Pleasingly or encouragingly, that significance goes away when you go to lower quality, lower confidence data sets, sorry, not quality, lower confidence data sets in the Safari database. So that's point one. And then point two, significant overlap on the genes that are differentially expressed in the samples, autopsy samples of autism individuals. We've tried to do the controls that we could think about. There's always more controls that one could imagine. We've done reciprocal controls. So we've looked at our socially unresponsive bee gene set, compared it to other human data sets that are relevant, mental illness related gene sets. We don't see any significant overlaps. We've done the converse. We've taken the autism gene sets that I just showed you and compared that to other bee behavioral sets, like as you can imagine, we've got a lot of them, and we don't see the overlap. So for now, we only see that particular overlap. Suggesting, again, not that bees are autistic, but that genes are involved in autism are ancient, some of them, have conserved roles, and can be involved in evolutionary independent instantiations of social behavior, and in particular, social responsiveness. So that's one point in this last part of the talk, and then here's the last point, and then we'll be wrapping up. So another way that we look at ancient conservation relates to the reward system and how the reward system could be involved in motivating social behavior. So I think we're all pretty comfortable with this construct here. How about this one? Might we go there? It's a little bit of a reach because people tend to think of insect brains, and rightly so, as perhaps simpler. And so do we need to invoke a reward system when we can think about a simpler construct like a stimulus response? But I think it's helpful to try, and I'll show you some data that I think support that notion. It's helpful to try because then we can kind of think about this. So if they're addicted to sweets, might they be addicted to altruism? These are all bona fide cases of the kind of products of an insect society. Language, manufacture, construction, warfare, and agriculture. So magnificent accomplishments. These are all traits that kind of change the face of the planet for better or for worse, especially in our hands. And they're all present in insect societies. They're all based on incredible altruism associated with the genetic structure of an insect society. And is it possible to imagine that one contribution to all of this is an addiction, where a manipulation of the reward system so that these behaviors are motivated? So I'll give you some information from our honeybee studies that supports this. And this now goes to the scouts who perform this waggle dance, which communicates information on distance, direction, and value of the food that they are looking at. Experiments with robots have shown that this communication system is about 50% effective at communicating. I've been married for 36 years. 50% communication efficiency is very good. So this is the system that the bees have. And so we asked the question of, when you see this kind of motivated behavior, might you imagine putting it in the context of the reward system? So what we did was we looked at one particular biogenic amine, a neurochemical called octopamine, closely related to dopamine. Many of you know dopamine colloquially as the pleasure juice of the brain. And the reason why we looked at octopamine was because experiments in Drosophila have shown, in the fruit fly, have shown that octopamine is involved in mediating food-seeking behavior and the response to food. And so that was a strong motivation. So we wanted to see whether this me substance, personal behavior in Drosophila, has been co-opted to structure dance behavior. Because one thing I forgot to say is that when a solitary organism finds good food, it eats more. When a honeybee finds good food, she does not eat more. She collects about the same amount, goes back to the hive, and dances more. So there's been a me to we transposition associated with what the organism does with the food. So the honeybee collects the food, barely eats any, brings it back to the hive, and dances more. So what we did is we took advantage of the fact that the dance language is a finely tuned system. That is, bees only dance if the food is really high quality. So, you know, like if you go out to an amazing restaurant, it's great, you're going to write a Yelp report and you'll tell your friends and so forth. If you find a terrible food source, you may do the same thing, but if it's so-so, you probably just won't say anything. So likewise, bees find a food source that's great, they will dance heavily for that, they will recruit for it. They find a marginal food source, they may use it themselves, go back and forth a little bit, but they're not going to dance for it. So what we did is we titrated using a large field enclosure that we have next to the bee lab, and we titrated the food reward, the sugar reward, so that the control bees were barely dancing. 10% of the bees were dancing. Then we jack them up with octopamine, and we can triple the number of bees that are dancing. So yes, they are using the octopamine signal and system, and that's manipulating their dance behavior. So to extend this, we took advantage of perhaps a little-known compound called cocaine and also extended this finding. So this is the 2 microgram octopamine. This is the cocaine treatment. Biochemically, I will share with you it's a trivial experiment because cocaine exists in nature as an insecticide to kill insects. The way it kills insects is it's an octopamine reuptake inhibitor. That is, it prevents octopamine from being removed from the connections between neurons, the so-called synapses, and then causing higher levels of octopamine neurotransmission. So the neurons never calm down, the insects twitch and shake, and they die. So that's what a cocaine exists. The reason why it may be known to some of you, and that's none of my business, is that it also interacts with the human dopamine system because cocaine and octopamine are so chemically similar. So from the biochemical perspective, this was a trivial experiment, but to push this reward concept, those of us that have done experiments know stranger things have happened, so we wanted to see whether it would behave as it was predicted to behave, and it was. And then finally, when we add an octopamine blocker, it also blocks the cocaine effect. So we could get bees to be 3 times more likely to dance to a marginal food source. So these are the scouts that are doing this dancing. The genes that distinguish the scouts, what we did this time wasn't a bottom-up approach, but it was a top-down approach. There's a literature on novelty seeking in vertebrates. Some genes have been implicated in humans, and we looked at that subset of genes, and we found that using that subset of genes, one could find strong differences in gene expression between scouts and non-scouts, sufficient to be able to distinguish most of the bees. Not a great pattern there, in part because there's so much genetic variation from colony to colony. But strong differences, and then something that I won't show you that really nails it, is that when we manipulate some of these systems using pharmacological treatment, we can turn non-scouts into scouts and scouts into non-scouts. So kind of closing the loop there, that these ancient, evolutionary conserved, reward system related compounds are involved in a different reward system. Again, making the following point that I want to emphasize here. So here we have the lineages leading up to the insect societies and lineages leading up to human society. The last common ancestor lived some 600 million years ago. Is thought to be a marine flatworm with a very, very primitive nervous system, not a centralized brain, no social life to speak of as far as we know. And the point for me showing this to you is for you to appreciate the context in which we're trying to put these results. So we're not saying that scouting is novelty seeking. We're not saying that social unresponsiveness is autism. We're saying that there's some genes that have common functions, and they can be used and reused in evolutionarily independent events of social behavior. And so think of it perhaps as the Ghostbusters model. So, you need particular molecular pathways to be involved in social behavior for whatever reason we don't know yet. Who are you going to call? You call these systems repeatedly in evolution. And that's an analogy that actually is now working better since Ghostbusters 2 came out. It was getting long in the tooth when it was only Ghostbusters 1, and people like us would know it, but anyways. So just to summarize, I want to come back to this slide, these last two slides that I showed you, but now with a little more information. Animal models are revealing the real relationship between genes and behavior. Using these examples, we see the dynamic genome in action across timescales. We see context dependence affecting gene expression. Some evidence for this, ancient co-opting of a system with scout bees addicted to altruism. There are other parallel stories in other species, wasps and voles, where you see basic molecular systems that have been co-opted. In voles, the oxytocin system, involved in various kinds of nurturing, including possibly romantic attachments. And so a lot of taking basic building blocks and then using them in different contexts. So one point is DNA is not destiny. And I want to also say just some very late-breaking work. Even DNA is not DNA. What do I mean by that? So a very provocative study that was just published in Science shows that early life experience can trigger, in mice, can trigger different behavior of elements, genetic elements called transposable elements that can insert themselves in the genome in different places and then give rise, really kind of play havoc with basic patterns of gene expression, or at least change them. So now this is not, all this stuff here is at the RNA level, this stuff here is at the DNA level. Environment, apparently, able to get in at the DNA level. One paper so far, the implications of this we don't know, but really very provocative. And then I'll go so far as to say, don't lose sleep. For those of you that saw Gattaca, a lot of strong evidence for multiple higher order interactions. A paper just came out in yeast showing 3 gene interactions with very unpredictable phenotypes. So it would take an amazing amount of knowledge and ability to even imagine being able to manipulate behavioral phenotypes in humans based on the data that are coming out from yeast. So where are we? We're poised for this new understanding of human nature. Brent Roberts has been taking traditional models of personality that are state trait models, using these evolutionary informed models from animals to be able to get at the idea that human personality traits are not either tabula rasa or biological, meaning set in stone, but this new model that we've been talking about today. And he's very interested in policy implications and has been discussing this with Professor Heckman as well. And then, of course, kind of the money points would be related to assessment and intervention in that context. The work here that was done here at University of Chicago, headed by John Cacioppo, basically saying it's kind of all in your head, that this study was notable not only because it was the first human study of a social trait, but the evidence for differences in gene expression, and by the way, these were in human leukocytes, blood cells, not brain for obvious reasons. The evidence, the connection was closer for self-reporting measures of loneliness rather than objective measures. So the individuals were assessed by social workers and by an objective measure. They also were self-reporting, and the connection was stronger for the self-reporting. And we've done a study like that as well. So bees are able to measure distance. As I mentioned, that's part of the dance language. We train bees to think they went a further distance by tricking them based on knowing how they measure distance. And bees thinking that they flew a longer distance showed the long distance dance pattern, gene expression pattern, rather than the short distance. I could tell you more about that. So kind of all in your head is also something that extends. The MAOA gene I mentioned has been introduced as evidence in court. So far, not during the trial phase, but only during the sentencing phase. Why do I know that? We teach at the Institute courses for professionals, genomics for judges, genomics for states attorneys, genomics for doctors, CEOs, and so forth. And so I learned about that by listening in. So the MAOA gene, because it has a genome by environment body of data to support it, has been brought in, please get my client a lesser sentence because of extenuating environmental circumstances. And then this paper here that I mentioned, but now I'll give a little more detail, really summarizes things very nicely. Brain and behavior develop over time. Genes are always expressed in context, and epigenetics is now seen as a mechanism to embed experience. And hopefully you saw some examples of that from the honeybee. So we're at a moment where we want to try to integrate this, so it's appropriate to reflect on the challenges that we have. We need better ways to measure brain gene expression in humans. If this is an important facet that needs to be added to the whole domain that we're trying to build here, then this is important. Right now we have autopsy or blood cells as markers. Might we think of ways of using brain organoids in new ways to get at environmental effects? Might there be new imaging methods? There was just a paper on disembodied pig brains. Might we be able to be using them in some way? I don't know, but the current two levels that we have do limit our progress. So it is something to think about. We're actually working on some imaging methods that I'd be happy to talk with any of you about. So let me thank the people whose work I spoke about. Hopefully I managed to mention most, if not all of them, some collaborators at the Institute as well as beyond, some of the technical genomic resources that we use, and our funding agencies, and thank you very much.
Thanks, Gene. Great lecture. We have to take questions, I'm assuming. We've got time.
Delighted to, yeah. Questions, comments?
How much do you think of like the social interactions is mediated by the role that we play? It's like the queen would have a need for constant interaction due to mating and getting fed, while a harvester bee might have much less need for social interaction because most of the work is relatively solitary.
Mm-hmm. Yeah, that's a fascinating question. Everyone hear it in the back? Bees that have different roles, some of them more connected to others, to other bees than other roles, how might that be manifest in their need for social interaction? Yeah, it's a fascinating question. I think we could actually test that kind of thing because they are really distinct roles, but we don't have any information on it now. That's a really cool question. Sarah. Hi.
I have sort of a big order question. So at the end, you presented some solutions that were, in order to bridge this gap in the interspecies collaborators. They seem to be technical solutions. And I'm wondering if some of the problem is actually intellectual limitations and lack of communication, and if you have ideas about sort of holdovers from behavioral genetics and how we can better, teach each other about commonalities and how to move together.
Yeah, I think that is very important, and I think it is part of it. So I'll have to admit, I wrote a couple pieces in 2004 to set the stage for this. It was a short piece in Science, and then I was asked to write an op-ed piece in the New York Times. And I say that on purpose to say that I thought, oh, okay, yeah, I'm done. This is now, the synthesis is going to happen. Didn't happen. And I think that the issue might be that, yeah, that may not have been contextualized right, and how to build those connections across disciplines probably takes a lot more work than just writing one thing and sitting back, and hence the visit here. Yeah, exactly. Yes?
You mentioned the comparison between novelty-seeking behavior and scouting, genes responsible for scouting bees. Were you able to do any kind of comparison to the human genotype similar to what you had done with autism, or?
It was more informal comparison, but in that spirit. And so the genes that we looked at where we saw the big differences between scouts and non-scouts, and which we used to structure the principal component analysis, those were genes that came by gleaning the literature for that. So the difference is that most of that work was still done under an older paradigm in behavioral genetics known as the candidate gene paradigm, which has largely been left behind. And the autism work has been more discovery-oriented, bottoms-up kind of thing.
I got a question.
Yes.
I'll take prerogative here. There's some great work coming out on the role of antimicrobials in affecting bee behavior. So some of Moran's work has suggested that there's a relationship between antimicrobial activity and dancing behavior. So the waggle dance gets knocked back or altered.
Yeah.
Would that, I mean, you probably call that under environment. You didn't say microbiome once in your talk, which was impressive. I'll give you that. But have you considered the interaction in that space? I mean, you know, if you took those animals, the bees that weren't interacting, and looked at their microbiome, would you expect to see differences?
So it's an area that we haven't gotten into as much as we should. We've done one study that compared nurses and foragers as two very different states where 40% of the gene expression in the brain is different and many different neurochemical, brain structural differences. This is all a setup to say that we didn't see any differences there. Now, we did this about 8 years ago. The methods, thanks to you and others, are way more sophisticated, so we really need to get back and do this. We have a collaboration with the Moran Lab to try to get this more instantiated, but there's a lot that can be done. It should be, based on what you see in other organisms. We were disappointed, but not shocked with our result because they are constantly exchanging material with each other. And so, you know, maybe there's a, colony level microbiome that is superseding a behavioral specific one. But it's a very rich area, again, to do the me to we stuff. See, so isolated bees compared to groups and so forth. Yeah. Yes?
For the bees that you saw that were reacting to both the intruder cue and the opportunity cue, was there any relationship with that and sort of plasticity of roles?
Yeah, that's a great question. We didn't study that. And so what we want to do is to be able to have our automated detection system running on all behaviors so that we could just say, that's a great question, and then just punch it in and get that information. So we're close enough to almost be able to taste what that would be like. But we're not there yet, so we have to do the lab assays that looked at those behaviors, and then we only have a couple behaviors automated so far. Yes?
Yeah, so how often do these bees change groups? And is it like on a scale? So do workers go to scouts or do scouts go to nurses? How's it shift?
Yeah, so I'll just give the context by repeating what I said before. They live for about 4 to 7 weeks, just to give you that background. And then they start working in the hive and then graduate and work outside the hive. So now I'll answer your question. Within the hive, the roles usually last a few days for each one, and then they move along. So they're a nurse for about 5 to 7 days. Then they're builders for another 4 to 6 days. Foraging usually is something that they do for the rest of their life. But then within some of these jobs, there's specialization. So they can become a scout or a recruit, but they're foraging continually. Then there's some other really weird jobs that they only do for about a day or so, but very intensely. So corpse removal is a job that only a few bees do, and they only do it for like a day or two, and they do it very, very intensely. So there's like highways of bee life, and then these bywaves of bee life, and strong individual specialization. We once studied a bee, Yellow 57, that was in our pre-barcode days, so colored number tags, that collected water from the same location for 17 days. Exactly the same location. It was a fun story on how we discovered, which I could tell you later, but it happened that way. So there's individual specialization layered on top of that. So an experiment we're thinking of doing by bringing together genome sequencing, transcriptome sequencing, and large-scale automated behavioral monitoring is a kind of a Truman story experiment, if those of you saw the movie. Constant monitoring of all that, having all this information to be able to put together these profiles. Yes?
Do you have any idea what prompts the transitions from role to role in the animal department? So,
Yeah.
I mean, what happened?
Yeah, so there is a set of hormones that kind of time their behavioral maturation, similar to how vertebrate hormones work to time sexual maturity. In this case, they're not maturing sexually because they don't do that sort of thing. But it's the same kind of thing where there are large-scale mechanisms that are timing their maturation. And it's a play on the solitary life. So what's nice about bees is it's easy to say something like that because there are some over 19,000 extant species of solitary bees, some of which have been studied, and so we know. So the solitary life is you emerge as an adult, you lay an egg, and then you forage and provision for it. So what you see in this division of labor is this lay an egg, and take care of it inside the nest, has been extensuated, and then the foraging phase has been extended as well. So, they don't have a bar mitzvah, that's right, but there is extended adolescence in the case. And that's, again, a feature of many societies where you extend periods of life that make the individuals more vulnerable and more connected to each other.
One more question, if there is one?
Yes?
For bees, is this a specialization, but is it pretty much species specific, or have you seen this specialization change in a meaningful way for one species as the environment changes? If you want one species putting around a different environment, and you solving see a new, entirely new kind of specialization develop or disappear.
So the general contours of these kinds of plasticity events or results can be seen, but the ecologies are very different, so then it gives a different context. So for example, ants are closely related to bees, and yet they don't fly, and so they're predators rather than vegans. And so how they go about making their nest and collecting their food is very different, but nevertheless they have very similar roles. They have foragers, they have individuals that live in the nest, they have guards, and so forth. How it plays out differs somewhat, but the broad strokes are the same.
Okay, thank you very much.
Sure.